Hi, everybody. I'm very sorry about my voice. This is not normally like this. So just want to check my clicker. Okay, perfect. So I'm going to talk to you a little bit about some work we're doing in this space, trying to really close some of the sensitivity gap we heard about yesterday in terms of when we do whole genome sequencing and when we do genotyping, we are not always able to predict with 100% sensitivity the drug resistance phenotype. And I'm going to talk about some, our approach to try to help close that gap. So as you heard before, there are really three main steps, the way I see it, that's going to link essentially, that's going to be this next phase of a drug resistance diagnostic. We want a test that's going to essentially go directly on sputum that will allow for generation, ideally, of the whole genome sequence. And then we need an algorithm that would take the whole genome sequence and make predictions, essentially, of resistance and sensitivity for the entire panel of available drugs for the patients. And then we need a method to immediately relay that information directly to clinicians, providers, laboratorians, so they can essentially act on it. So you heard a little bit about this yesterday in the session. I think you'll hear more about this kind of phase of going sputum to sequencing or sequence data tomorrow. And we heard a little bit about how you would, what kind of information you need and how quickly you need it a little earlier today. What I'm going to talk about is kind of this interface where you go from the data, how you actually interpret the data to a resistance phenotype. So we all know that there are actually several genes and mutations within that have been clearly associated with drug resistance. And the dogma in TB genomics is that for the most part, a strain that doesn't have a mutation or is so-called wild type is going to have essentially a very low MIC, is going to be sensitive. And then when under positive selection, it acquires, for the most part, one mutation, then quickly, rapidly elevates the MIC to a high level above the testing concentration, and then you have resistance. And so for most part, the dogma is all you need is one mutation that would elevate your MIC to a clinically meaningful level. However, the reality is more complex than that. Whereas it is true that there are certain mutations that elevate your MIC alone to high levels, and there are notable examples of this, but it's also true that this is not the case across the board. So I'm going to give you a few examples of this. So this is a series of samples that were obtained from the same patient over the span of 10 years. And this is the INH MIC. And you can see that over the span of 10 years, so roughly speaking, these were sampled annually. In this patient, actually, it was reported on back in 2005. This was a patient from Italy. And you can see that there were actually several step-ups in the MIC for this patient. And when they had done Sanger sequencing at the time of several of the canonical genes, CAT-G, HPC, a few other INHA and INHA promoter, the only mutations they could identify by Sanger was this CAT-G234 mutation and an AHBC promoter mutation, which didn't actually really quite coincide with these step-ups. So the question is, what else happened for this strain that explains this resistance? And there's probably mutations outside of these regions or mutations they were not able to detect using their technology that would explain these. So it's not a one mutation, one resistance phenomenon. And in vitro, so this is data from David Allan's group. This is kind of old data now from 2008. So they had actually constructed ethambutol resistance mutant. They added the EMBB 306 variant in vitro, and they essentially tested isogenic strains, one with the mutation, one without. They showed a kind of a variety of ethambutol MICs depending on the strain background. So you see that there's variation and interaction with the strain background. But on top of this, just introducing this EMBB mutation, which is in a gene that's really not supposed to affect isoniazid resistance or rifampicin resistance, but supposed to cause ethambutol resistance. And when you actually expose those strains to low concentrations of these other drugs, there is a clear survival advantage in these low MIC levels. So there seems to be clearly interactions between one mutations leading to survival advantages for different kinds of drugs. So this was observed for both INH. So you see the separation here between the open graph and the closed graph. This one had the mutant, this one didn't. The mutation, sorry, and this one had the mutation, this one didn't. These were increasing concentrations. And similarly, that was seen for one concentration of rifampicin. Again, arguing for kind of a higher complexity of resistance. In addition, David Lalonde's group produced a paper several years later where they mutated several genes within the pathway that's targeted by ethambutol and showed, in fact, that there are cumulative mutation accumulations leading to step-ups in the MIC. And so what's emerging is this kind of situation where you have really interactions between mutations that's actually eventually resulting in the observed MIC in the strain. And this is sort of evidence from different data sets. Again, this is for rifampicin, where you have this RPOB, this is the E. coli numbering, Q513L introduced into 3 different strains of 3 different backgrounds. You can see that even though all 3 of them are resistant, the MIC range is really incredibly different across these strains, also arguing for interactions. And further, that our experience with fluoroquinolone resistance is that, in fact, the higher the MIC, the more likely you are to see mutations in more than one gene. This is a small study, but this has been also an observation that we've made. So to kind of summarize this, or really summarize this, this whole question of how do we actually then think about this molecular diagnostic gap for both sensitivity and specificity. So for sensitivity, there are several possible explanations, and it might be kind of a combination of these that's actually happening. One, it's possible that in fact, some of the phenotypic tests are overcalling resistance. And so, in fact, whatever percentage of strains that are actually being called resistant by phenotype will never have mutations. And that's, it's possible. That being said, I don't think that that explains probably the entire gap for some of the second-line drugs, for example, pyrazinamide, because we know that there is evidence from retrospective studies and other meta-analyses that, in fact, the DST or the phenotypic DST accurately predicts treatment response or reliably predicts treatment response. So even though there is probably some phenotypic overcall, I don't think this is likely to explain the entire picture. And this also, you heard yesterday about this phenomenon of heteroresistance. I call it heterogeneity, whereby you, when you essentially do sequencing on pools from culture, you're likely to see some subpopulations of isolates that have mutations, whereas the entire population doesn't always have that same mutation. And so when you have these minority populations, that could explain the discrepancy if you're not picking those up. There's also this other possibility that, in fact, we just don't know all of the mutations that are relevant, and that we don't know all the genes that are relevant. There, in fact, may be mutations outside of these regions. And this phenomenon that I was just alluding to, this epistasis or gene-gene interaction phenomenon that we have really not studied at all up to this point. And even though you've heard a lot that the specificity is not a problem, the reality is, I think that if we are seeing perfect specificity, we're probably not including enough mutations, because we know for a fact that there are, for example, for drugs like ethambutol, mutations that do cause MICs that are very close to the critical concentration. So the fact that we're not actually including those potentially in our screens as predictive of resistance will end up with perfect specificity, but the reality is we probably need to include them, and these kind of intermediate MICs are going to come up and they're going to limit the specificity too. And so, a possible explanation, other explanations for kind of a lower specificity could include the same factors like epistasis, sort of the reverse issue of phenotypic undercall, but also false positive associations that you might make if you don't include sufficient number of drug-sensitive isolates in your study or survey. So I'm going to talk to you a little bit about a study that we had done, and then also talk about some follow-up work that we're doing. And so in this study, which was originally funded by the Gates Foundation, what was essentially done, there were 7 national, supranational reference laboratories that contributed a large number of mostly MDR and XDR isolates. Initially the plan was to sequence 1,800 isolates. The plan was initially to do Sanger sequencing, but the cost was actually prohibitive at the time this was done, about 5 years ago now. Essentially, what we ended up doing is actually a targeted type of technology, not too different from what was described yesterday, but not using amplicons, but using molecular inversion probes. And so, this was done on 28 different genes and intergenic regions, and these were essentially a summary of where all the strains came from. And when you actually collect strains from many different settings, especially many strains from the same lab that are drug resistant, because of the amount of transmission of drug resistance that's actually happening, you want to confirm that in fact the strains are independent strains to the extent possible, that they weren't from transmission strains, that they weren't very closely associated with each other. And you need to confirm this in order for you to essentially be able to then go on to the next step and associate the genetic variants that you see in them with the phenotype. Because if you don't correct for this potential similarity in their lineage or in their kind of background, you are, this is called an issue with population structure, and you might then be more likely to make false positive associations between, neutral variants that are lineage markers with resistance phenotype. So you have to confirm this, and that's why we constructed this phylogeny, and we looked at the distribution of sensitive and resistant isolates within this phylogeny, and in fact, the sample was balanced. So we proceeded to do the next step, which was construct a prediction model using the genotypic information. I'm glossing over a lot of details, so I'm sorry, but if you have any questions, I'm happy to answer later. So here we decided that we would take kind of, arguably a very complex approach to trying to associate the genotype to phenotype. And we wanted to take into account this issue of gene-gene interactions in our model to some extent. But also we wanted to learn to the extent possible from the data. We didn't want to go in, with all of our priors and then say, okay, I know these are the relevant mutations. I just want to see, how many, how they predict the resistance in these strains. I want to actually learn what are the relevant mutations from this strain set, because to that day, at that moment, this was the largest number of drug-resistant isolates that were ever studied in this way. And we had actually over 1,000 multi-drug resistant isolates. So we decided to use this method called random forests, and essentially what it does is it takes in the mutations we restricted by gene for each drug. We kind of didn't associate everything with everything, but we restricted by gene, but looked at everything within those genes. And then constructed a prediction model for each drug, and then on 2/3 of the data, and then we validated it on 1/3 of the data set. And what we wanted to also do was essentially make an assessment as to minimum number of mutations that you need to make accurate predictions. So we constructed, what we did was we bootstrapped this whole procedure. So we construct the model. We then essentially remove the least important variable, and you can do this with random forest because it nicely classifies variables by importance, by predictive importance. And then we rerun the model after removing the least important variable. And when we do it again, we remove the next least important, and so forth. And we did this sort of 100 times for each of the drugs and kind of summarize the data. And this is how you get this, essentially, these plots. Essentially, these are sort of ROC-esque plots where you can see that, in fact, we had put into the model 220 mutations for isoniazid, and in fact, the grand majority of them are not helpful for the prediction. When you remove them, the sensitivity and specificity, stays completely flat. And so until you reach this kind of lowest 20 or so mutations, I think there were 18, and when you start to remove mutations at that point, you start to see a drop in your sensitivity. We did this also for rifampin, and you can see something similar for rifampin. In fact, you need less mutations. Oh, and we know this is the case for rifampin 9H because there are reasonably accurate commercial-based tests for these drugs. But for pyrazinamide, that wasn't the case. In fact, you needed almost all of the mutations to make accurate predictions. It was actually fairly unstable. Like each of these mutations that we saw were individually rare, but they were actually highly, highly predictive in those strains. There was simply no other mutations, and they almost never occurred in sensitive isolates. So the automated procedure actually kept them. So this is the final kind of results here. These are the validated diagnostic performance. And so you'll see that, and I should also mention, sorry, that we sequenced all of these genes, and I didn't give you an entire list of them, but for example, we sequenced all of PNCA and its promoter regions, and actually the flanking 100 base pair after the gene. And we also not only assessed for SNPs, but we also assessed for insertions and deletions. And so this kind of summarizes all of this. So even with that, sequencing the entire open reading frame and the promoter, in fact, our sensitivity for pyrazinamide was still in the validated was still in the 60% range. On the training data set, it was around 70, but when we validated it on the independent data set, it was in the 60% range. And so you can see here actually the total number of mutations that were important for the prediction. You see that those are small for isoniazid and rifampin, very large for pyrazinamide, and kind of all over the place for some of the other drugs. And then you'll also see that for ethambutol, even though we forced the model to give us very high specificity, it was just not possible for us, for the model to generate a specificity that was very high. And I think this is because of this issue of these mutations actually causing intermediate levels of MICs. So we've now, and I'll talk about this this afternoon, embedded this model in an interface that allows users to directly input their whole genome sequencing data and get a prediction. And we invite everybody to use it, and I'll talk more about it a little later. So I went ahead and actually, and I have to say, I want to thank Marco for inviting me. I forgot to thank him earlier. And also, they've really, and Reseek TB, they've really been quite incredible in reaching out and wanting to collaborate on this. And I can't thank them enough. They, in fact, gave us the public portion of the Reseek data set, and we've been working with them to try to validate our prediction model using the Reseek data set. And we actually, so we have actually now downloaded and the FASTQ files for 2429 isolates, where I think we're missing a few relative to the Reseek. I think there were issues with either getting the data set. We were actually downloading it from NCBI and EBI, but now I know I can actually download it from Reseek directly. And also called variants using our own pipeline, so I wanted to test our pipeline end to end, really. And in the process of doing that, I actually have since refined my variant calling algorithm because I realized that I was being probably a little too stringent relative to the variant calling pipeline that they've used. So that's actually already helped us improve the algorithm. But after doing this and then predicting resistance using the random forest method that was trained on this other data set. I haven't retrained the prediction model yet, but you can see here that overall the sensitivity and specificity is pretty comparable across the board, except for these ethambutol, pyrazinamide, and streptomycin, where essentially the RF prediction method actually, or the random forest method, does have a significant advantage where the sensitivity is about 10 to 15% higher compared to essentially using a list of canonical variants that we obtained from again, there's so many different possible lists of canonical variables. What we actually ended up using was what's currently published on the ReSeq site. I know that's constantly changing, but I needed some kind of point of reference. So, and this I think hopefully argues for potentially the advantage of these more complex models. And I know that ReSeq is also interested in how these models can work and whether or not they have an advantage over more traditional methods. I also wanted to sort of observe in passing that on this data set, again, this was sort of a subset of what's currently published, because I know that there are non-public strains that are currently summarized on the ReSeq website. But it's notable that a big proportion of these are monoisonazid resistant strains, which up to that point we actually hadn't worked with that many monoisonazid resistant strains. And in fact, the sensitivity of both our model and the canonical mutations for predicting resistance in mono-INH resistant strains is significantly lower. It's in the 80s. It seems to be in the 80%. And this is an interesting observation, whereas you don't see as many catchy S315T mutations and INHA promoter mutations as you see in multi-drug resistant strains, which also argues that potentially this kind of early form of resistance might be more complex. You might have potentially slightly different mechanisms. Next steps after this, so what we're planning to do is we're going to actually be retraining a prediction model on using a pooled data set, our data and actually the ReSeq data and actually additional public data that's now available from the Broad Institute. But what we're planning to do is also use a different algorithm than random forest. What we've been working with is a neural network model, and we might even branch further into kind of deep learning. And just to give you a little flavor of how the performance compares. So just comparing on the old data set, all training data, comparing random forest to neural networks. So you'll see here that there's number 1 and number 2. Number 2 involves also adding variants that only occur in sensitive isolates. So previously we had excluded those, thinking, mutations that only occur in sensitive isolates are unlikely to be of value when you're making a prediction. But it turns out actually it does help, at least in the training context. We'll see once we validate it on additional data. But if you look at these 2 lines for now, you see that just using a different algorithm, different predictive algorithm, same data input, that the overall AUC improves and the sensitivity improves by about 10%. Specificity is actually a little better. It doesn't get you don't lose significantly. And so I think that there's also, this is a further argument that using these more complex models on clean data sets where you don't have significant confounding is going to really help you make better predictions and hopefully close the sensitivity gap. And just to kind of, sorry, I think I lost a slide here, but that's all right. I think I'll talk about it a little later, and I'm almost out of time. thank you all. This is my acknowledgement slide, and there's quite a few people who have contributed to this work, and I wanted to also thank the funders that have supported us over the years.